to our coffee chat, focusing on the role of the microbiome in cancer diagnostics, exploring current challenges and future hopes for the field. I'm Jade Parker, Senior Editor of Oncology Central, and I'm delighted to be joined by three leading experts in microbiome oncology research. Eddie Adams, Chief Scientific Officer of Micronoma, Greg Spitch-Poore, Chief Analytics Officer, and Sandrine Miller-Montgomery, Chief Executive Officer. Firstly, we'll start off with some introductions. Eddie, would you like to start by introducing yourself?
Sure, my pleasure. My name is Eddie Adams. I'm the Chief Scientific Officer at Micronoma. My formal training is in biochemistry from the Massachusetts Institute of Technology. And I've largely been an industrial scientist throughout my career, really focusing on technology development, technology platforms, predominantly in diagnostics, but also earlier in my career, I worked extensively in nanotechnology. When I learned of the amazing work being done at the Center for Microbiome Innovations at UCSD by Greg and the team there, I absolutely had to be a part of that. And that is what is now Micronoma, where I've been for 2 years.
My name is Greg Spitch-Poore. I'm the co-founder and chief analytic officer of Micronoma. This field is really personal to me. I lost my grandmother due to a very, very late stage diagnosis of pancreatic cancer as a freshman at college. And ever since then, I've been trying to create better cancer diagnostics. In particular, for her case, she was diagnosed in late stage metastatic disease. She failed therapy only after about 2 to 3 weeks. And given that there was no benefit to the therapy that she was being given, and yet there were many side effects, she had to discontinue therapy and passed away from the disease just 33 days later. I went to medical school and decided that I was going to become a physician scientist specifically to create these better cancer diagnostics. And I was surprised that when I arrived at medical school, I discovered just how much room there was for improvement. And despite all of the research that's happened in the last 50 years, the 5-year survival rate for pancreatic cancer really has not improved that much. In fact, it's just peaked above 10%. Then as a second-year med student, I was very surprised to see a study that was published in Science by Ravit Straussman's group, who's now on our scientific advisory board, discovering that more than 75% of pancreatic cancer patients actually had bacteria inside of their tumors. And many of these bacteria were able to degrade directly gemcitabine, which is the gold standard chemotherapy being given to these patients. And it really struck close to home and suggested that perhaps we were missing a factor that was really important in both the diagnosis and care of patients with cancer. And so I devoted my entire PhD to study the presence and role of microbes in cancer, which to date has now been published in Nature, Science, and Cell. So my research is a founding principle for Micronoma, and I've been very fortunate to find such wonderful co-founders and colleagues to pursue this effort with me.
Thank you, Greg. Sandrine, could you introduce yourself for us?
Sure. So I'm Sandrine Miller-Montgomery. By training, I'm a scientist. I'm a PharmD and a PhD in medical science. I have had the opportunity to stumble in the field of microbiome a little over 10 years ago now. And I was trained in the human science aspect of biology and was mind blown by all of this science on the microbiome side that we have been ignored. And I decided that it was a field I wanted to pursue. Along the way in my career, I moved from scientist, bench scientist, R&D manager to more, as Eddie mentioned earlier, doing more the business side, the sales and the marketing of life science company. At some point, I was able to join Rob Knight at UC San Diego to help him grow and co-found and grow the Center for Microbiome Innovation. And that's during that 3 years in academia that I met Greg, who came to us with this amazing project he just described, and this fire to discover a solution for cancer detection and more than that in oncology. At some point, Greg, myself, and Rob decided that, okay, we needed to put that discovery in the hand of the clinicians because we all have our stories around losing people around us, or people deeply affected by cancer, and we're trying to make a stop to that. as fast as we can. And so we co-founded Micronoma in 2019.
Thank you all. So to kick off this coffee chat, let's start with the fundamentals. So as you may know, the microbiome is a rich community of all the microbes, so bacteria, fungi, protozoa, and viruses that live both on and inside the human body. It's been said that there are more microbes in the human body than there are stars in the sky. However daunting of a task, if analysed properly, the microbiome can reveal the status of our health. So microbes are known to be found in human tissues, including cancer tissues and blood, which brings us to our talk here today. So let's start. So Greg, what is the circulating microbiome and why is it so promising for early cancer detection?
That's a great question. And I think I'll start less so with the circulating microbiome and more so just with circulating DNA in general, because I think that's what most people are familiar with. It's been appreciated for many, many years that human tissues release human DNA into the bloodstream as cells turn over. Sometimes this is a good thing. It's a good process. For example, we can detect fetal DNA as a fetus is growing within a pregnant mother. And we can use that as a non-invasive test for fetal chromosomal abnormalities, for example. And that has launched a whole new field of diagnostics. But this can also be used for the detection of cancer when something that is malignant is growing, particularly in a tissue that's growing rapidly and turning over rapidly. And so there's been a wealth of diagnostics in the last 10 years or so that have been focused on trying to detect fragments of tumor DNA that's being shed into the bloodstream. And so analogously, the last 3 to 5 years or so have showed that microbes exist and live, they're metabolically active within tumors, and that each cancer type has a distinct community of microbes that live inside that particular cancer type. And so what this means is that as these microbes are living and thriving within the tumors, they're also turning over and they're shedding their DNA into the bloodstream. And this is in small quantities, but it's still sufficient quantities for us to be able to detect. And so what this microbial DNA, as it's being shed into circulation, allows us to do is detect the presence of a cancer, but also the type of a cancer. And that's really why the circulating microbiome is such a powerful tool for cancer diagnostics.
And then can you talk us through your recent publication on profiling fungal communities in several cancer types?
But this story really goes back to our first paper, which was published in Nature in 2020, in which we profiled the Cancer Genome Atlas's compendium of whole genome sequencing and RNA sequencing studies. For context, this was more than 18,000 samples that we analyzed across more than 10,000 patients in more than 30 different cancer types. So it's an absolute wealth of data. In fact, just the sequencing data alone comprised roughly 600 terabytes worth of information. And we took all of that data and instead of doing what most people had done, which is using the human information to analyze various aspects about cancer, we threw away all the human DNA and RNA and instead looked at all of the non-human DNA and RNA, which is defined as it just simply didn't align to a human reference genome. And within that set of DNA, we started looking for pieces of microbial DNA. And in fact, we found a lot more than we anticipated. So somewhere between 2 to 3% on average of the whole sequencing data was microbial, meaning that since we had 6 trillion reads in the Cancer Genome Atlas to begin with, we literally had billions of microbial DNA and RNA reads at the end of this. And that analysis was mostly focusing on bacteria and viruses across cancer tissues, as well as matched blood samples from these same patients. And the surprising thing that we identified in that study is first that no human cancer type that we analyzed was sterile. And the second thing that we found is that you were able to distinguish a given sample's cancer type, whether it belonged to tissue or to blood, simply by looking at the microbial information within that particular sample. And related to this, the third major conclusion of that study is that we could actually use blood samples in order to distinguish which patient had a given cancer type. So that was most of the main conclusions that we made in our 2020 Nature paper. And the fungal story is really a second chapter of that paper. I would say that a lot of people were very interested in the role of fungi because virtually everywhere in the world that we look and find bacteria, we also find fungi. and very often they're interacting with each other. But prior to this past year, it had been really difficult in order to analyze the genomes of fungi because they're much more similar to human DNA. After all, they're both eukaryotes, fungi and humans. And that means you have to be much more careful in how you're filtering out your DNA to make sure that you're not getting human reads that are being misclassified as fungi. And so we worked really hard on these methods to be able to accurately classify when we had a piece of DNA that actually indeed was fungi to call it as such, and when it was human to call it as such. And we took all the information that we had developed for our 2020 Nature paper, and we reprocessed it using better methods and newer databases, which allowed us to characterize all of the fungi present within those samples, in addition to bacteria that we had been previously characterizing. And what was really surprising was that the fungi completely replicated the story of bacteria, even though they were present in much lower abundances. So if you look at the literature, roughly about 1% of all cells in a tumor are bacterial, based off of qPCR estimates. And if you look at the ratio of bacteria to fungi, roughly for about every 100 bacteria, you get about 1 fungi. So it's 100-fold less than the bacteria, which was to begin with only about 1% of the tumor. So to find that we were able to distinguish which cancer type a given patient has solely using fungal information was just really, really surprising. And we ended up partnering with another institute, Ravit Straussman's lab at the Weizmann Institute, in order to start doing meta-analyses effectively. So we took the Cancer Genome Atlas's compendium of data and fungi data that we had profiled, and we compared it to what the Weizmann Institute had found in their own completely independent 1,000-patient cohort. And we ended up finding very, very similar conclusions, despite these patients not being related to each other. And I think this simply substantiated the veracity of the findings. And I'll say that the one last thing I want to mention here is that as part of this study, we took a completely independent data set that had been previously published by a lab at Johns Hopkins and published in Nature back in 2019, specifically looking for fragment sizes in plasma in order to use those fragment sizes as a way to diagnose cancer earlier. But when we took this data set, which again was processed independently of us, and we profiled it for microbes, we were very surprised that we could actually diagnose cancer versus non-cancer individuals across 8 different cancer types with actually better diagnostic performance than what the human fragments were capable of diagnosing. And this observation held even when we subset our analyses only to tissue-resident bacteria and fungi that had been identified by the Weizmann Institute. And all of these conclusions also were still maintained after stringently decontaminating and implementing this decontamination procedure based off of roughly 300 experimental contamination controls that the Weizmann had run. So it was just a really, really phenomenal set of findings, and we were very happy to be able to release this into the public to show the utility of the cancer microbiome, and particularly the plasma microbiome, for diagnosing cancer earlier.
So, Eddie, could you tell us, how does a microbiome-driven biopsy compare to traditional ctDNA liquid biopsies?
Yeah, certainly. So traditional ctDNA-based liquid biopsies are based on the detection of tumor-derived genetic mutations that have been shed from the tumor during cellular turnover, and in some cases through active secretion mechanisms, such as, say, through the production of exosomes. Although tumors are composed of cells that have escaped traditional or canonical cell death pathways and resist cell death, there is some level of cancer cell death in a tumor. And much like a normal cell, when cancer cells are undergoing cell death, there is an enzymatic fragmentation of the genome. So you basically have the genome of the cancer cell cut up into very small pieces, which can escape into circulation, where they can be isolated through standard DNA extraction techniques for downstream analysis. However, because ctDNA, it requires cell death or secretion by a tumor. Its quantity in circulation essentially is a function of tumor size, which is to say that in early stage cancers or early stage tumors, you find very low quantities of circulating ctDNA as opposed to more advanced cancers where the frequency of mutant alleles of a particular cancer-associated gene to wild-type alleles is much less. Traditional ctDNA-based liquid biopsy assays have some great selling points. One both detects the presence of a cancer while at the same time learning something about the mutational status of the tumor. There are notable downsides. First, ctDNA assays are absolutely dependent on the amount of fragmented tumor DNA, as I just mentioned, released into circulation by a particular cancer type. And again, this means that late-stage cancers, say stages 3 and 4, are more readily detected by ctDNA-based assays than stage 1 and stage 2 cancers, whose decreased cellularity leads to much less ctDNA release. So this lack of sensitivity for early stage cancers, for early cancer stages, means that cancers are most frequently detected when they've progressed to a point where surgical intervention may no longer be an option. Secondly, the specificity of ctDNA assays can be significantly hampered by a physiological process known as clonal hematopoiesis. Clonal hematopoiesis is a natural process whereby white blood cells in circulation accumulate genetic mutations. And as many of these mutations occur in the same genes that are detected by ctDNA assays, it essentially creates this signal to noise issue in which it becomes very difficult to determine if the mutations are truly tumor derived or if they actually just owe their origins to white blood cells. For example, there was a really important study in 2019 that came out of the Memorial Sloan Kettering Cancer Center, where the researchers sought to determine the origins of ctDNA using ultra-deep sequencing, where they sequenced cell-free DNA to a depth of 60,000-fold. And they looked at, in the study, they looked at 508 cancer-associated genes. So they sequenced ctDNA from 124 patients with metastatic cancers, along with genomic DNA from matched patient white blood cells and where available, normal tissue from the same organ as the metastatic cancer in question. And what was amazing is they found that as much as 53% of the mutations found in ctDNA of cancer patients were also found in their respectively matched white blood cell compartment. So this was a very, very clear indication that the mutational contribution from clonal hematopoiesis is extremely significant and therefore tremendously decreases the sensitivity of ctDNA-based assays. So although that paper and others have suggested some control measures one might be able to take to correct for this signal to noise issue, it's not entirely clear to what extent those control measures are implemented in commercially available or commercially implemented ctDNA-based assays. In contrast to these assays, microbiome-driven liquid biopsy assays use features, cell-free microbial DNA, cell-free microbial nucleic acids that are completely independent of the cancer genome and therefore are not intrinsically limited by cancer stage. And again, because they are not dependent upon the detection of cancer-associated mutations, they allow us to completely sidestep this troubling issue of clonal hematopoiesis. So we're not restrained by looking at those mutations. We can employ analysis of those mutations in concert with the microbiome. However, again, using strictly microbiome signatures allows us to get away from this specificity issue imposed by clonal hematopoiesis. Another important point is that I alluded to earlier is that particularly in early stage cancers, where the frequency of mutant alleles to wild type alleles is so low. Contrast, microbiome-based assays or in the microbiome-based assays that we've been developing, where we use shotgun metagenomic sequencing of all cell-free DNA in a sample, we actually find that there is a tremendous amount of signal present, where typically, routinely, 1 to as much as 4% of the DNA signal in a cell-free DNA sample is actually of microbial origin. So this gives us a huge reservoir of microbial features that we can identify, and through the application of machine learning, arrive at very complex, high complexity microbial signatures.
In fact, if you don't mind, maybe, Greg, could you share your imagery around multiple shot on goal? Because I found that very telling about the power of why microbiome is helping us to be more sensitive.
The analogy that I like to use is if you asked LeBron James in order to take basketball shots, but from across the court. If you only gave LeBron one shot, it's possible that he would make it in, just because LeBron is that good. But if you were to give him 1,000 opportunities in order to shoot from the other side of the court, it's very likely he would make at least one of those in. And this is an analogy to how the microbiome allows us to enhance the sensitivity of our test. So in early stage disease, something that's important to realize is that there's very few driver mutations that are available to detect, usually less than 5 altogether. And it's possible in later stage disease that you're going to have an accumulation of driver mutations and other chromosomal events that you can detect in the bloodstream. But in early stage disease, there's just really not that many markers in order to use. And the difference with the microbiome is that we're working with hundreds to low thousands of markers, even in early stage disease. And so instead of having a few attempts on goal, we're now having hundreds to low thousands of attempts. And we really only need a single biomarker in order to confidently change between an individual who's healthy versus an individual who has cancer in order to be able to correctly diagnose that particular individual. And so we think that's what lends the high sensitivity of a microbiome augmented cancer diagnostic test.
And then just to kind of lead on from that, so where are the potential sources of microbial information in the tissue and plasma coming from?
Yeah, this is a great question, and I get it all the time, and so it's completely understandable. I think the right way to think about this is that we, just from the human angle, we would consider a breast cancer to be phenotypically distinct from a colon cancer, which would be phenotypically distinct from a lung cancer. And there's lots of research on why these cancer types have some similarities and also some major differences. But I think one thing to dramatically consider is that they're anatomically in different regions of the body, and that's going to lead to a very different microenvironment. And so the way that microbes are going to get to a particular tumor is going to depend upon which pathways are available for them to move, which is physically move from one location in the body to the next. But two, it's going to depend on the kind of microenvironment that those cancer types are fostering. And I'll just give a few examples. So for example, the case of pancreatic cancer, there's been some interesting mouse studies that have been led by Florencia McAllister's lab at MD Anderson. And what they were able to do is directly compare the ratio or the percentage similarity of microbes that were found in the gut of mice versus microbes that were found in the pancreatic tumors of those same mice. And it's important to note here that the pancreas is located adjacent to the GI tract and has a direct portal through the pancreatic duct. And so in this case, there was as much as a 40% overlap between the microbes that were present within the GI tract versus those that were in the pancreas. But that sort of pathway doesn't necessarily make sense when you're considering how microbes may get to breast cancer, for example. And simply because it's in a different region of the body, it has a different microenvironment, there's going to have to be different pathways. And I really like for this example to point to Fusobacterium nucleatum, which is perhaps the second best studied bacteria related to cancer other than Helicobacter pylori. And Fusobacterium nucleatum, although it's still debated whether it's a true commensal or not, it's often found in the oral cavity. And it was first originally discovered to be highly associated with colorectal cancer. And I think most people had assumed that, well, Fusobacterium nucleatum is found in the oral cavity, it just must make its way down the GI tract in order to eventually infect colorectal cancer. But there were some features of Fusobacterium nucleatum that were surprising, that didn't exactly behave the way that we thought that this hypothesis was valid. And so people did some really great experimental work, and they ended up identifying that Fusobacterium nucleatum has a particular receptor called FAP2, which adheres very strongly to a lectin called GAL-GALNAC that colorectal cancer happens to overexpress. But it's interesting because breast cancer also just so happens to overexpress GAL-GALNAC in many cases. And what do you know? We end up finding that many breast cancers have high concentrations of Fusobacterium nucleatum. And so what all this experimental data has shown over the last 3 to 4 years or so now is that Fusobacterium nucleatum very likely does not transit through the GI tract in order to get to colorectal cancer. It actually transits most likely through circulation. This is true for colorectal cancer, and it's also true for breast cancer. And so the bloodstream kind of becomes a highway of sorts in order for one bacteria to translocate to the next. I need to make it clear, just so that everyone's aware, that we're not talking about thousands to millions of bacteria moving through the bloodstream at once. Like, these are self-replicating entities. And it may sound scary when you consider it, but you really just need a single bacteria to home to a place which they would call home, right? And if you have a single Fusobacterium nucleatum that's able to home to a colorectal cancer, if it's able to adapt to those conditions, well, that single bacterium can multiply and divide and eventually become a community of Fusobacterium nucleatum living within the colorectal cancer. And the better the conditions are for that particular microbe, the more that it's going to thrive within that particular environment. And I think we really have a long way to go in trying to characterize all the possible mechanisms by which a microbe is going to get to a particular tissue. Again, Fusobacterium nucleatum is the second best studied bacteria associated to cancer, and we've just begun to characterize some of these mechanisms. But I will say that from our standpoint, with respect to the circulating microbiome, we've done many analyses in which we've taken standard reference databases and mapped all of the non-human DNA that we're identifying against these microbial reference databases. And we'll find somewhere in the hundreds to low thousands of hits of DNA reads that end up mapping to these microbes, even when accounting for potential contamination based off of controls that we're running. But if we subset these analyses to bacteria and fungi that were only identified in tissue by an external collaborator group, so this is Ravit Strassman's group at the Weizmann Institute, and collectively for bacteria and fungi, they've run over 1,100 experimental contamination controls. So we're very certain that these are real bacteria and fungi that are present in cancer tissues, but in a completely independent cohort. So if we subset our blood-related analyses to those tissue-derived features, we end up finding that our classification performance is nearly identical, whether we use just that subset of bacteria and fungi, or if we use the broader database that we've applied. And what's amazing about this is not just the fact that they're tissue derived, but they're also, it's a substantially smaller feature set. So it's roughly 25-fold smaller to use just the bacteria and fungi that overlap with the Weizmann data sets in comparison to the full databases that one could apply, and still retaining the same diagnostic performance, which inherently suggests that the bacteria and fungi that are most important for diagnostics are really coming from the tissues themselves, and specifically the tumor tissues in the case of a cancer-bearing individual. So again, I think it's a great question. I think there's a lot more research to be done in this area to characterize every specific mechanism. But I really think our data and the field's data has suggested that microbes residing within tumors is a major source of circulating microbial DNA that we're detecting.
Thank you, Greg, for talking us through sort of the classification and characterization according to different tumor types. So kind of leading on from that, so Eddie, could you talk us through how does a microbiome-driven biopsy fit in with the current standard of care for lung cancer diagnostics?
So it's been estimated that in the United States alone, every year, approximately 1.5 million pulmonary nodules are incidentally detected. Say a patient goes in for a chest X-ray for any number of reasons, and a nodule is detected in the lung. In addition to these incidentally detected nodules, there are a large number of nodules detected through lung cancer screening efforts in which patients with a history of smoking undergo imaging of the lungs to identify the presence of potential cancerous tumors. And so when we entered into developing a liquid biopsy assay for lung cancer, we looked at this space and asked the question, well, what is a tool that would really help clinicians? We see an opportunity to develop a liquid biopsy assay that doesn't necessarily screen an otherwise healthy individual for the presence of lung cancer, although longer term, we could in fact enter that space, but rather to address this key clinical question, which is, given the presence of a pulmonary nodule, whether it was incidentally detected or detected in a lung cancer screening environment, what is the status of that nodule? Is it malignant or is it benign? Pulmonary nodule management is an extremely complex endeavor that requires very dedicated multidisciplinary teams. It requires a lot of longitudinal analysis. And so we thought this was an excellent opportunity to provide a tool to the existing standard of care where, for the lack of a better phrase, a big portion of a big part of the current standard of care, which is imaging based, is a sort of a wait and see approach. If you detect a nodule that is below 3 centimeters, 3 centimeters sort of being a triggering point for immediate removal through surgery. The current standard of care is basically to wait and to continue to monitor that nodule over time. Do you see that the nodule is growing 6 months later? If so, then perhaps you might take surgical action or other options that exist. And we feel that given the speed at which tumors can divide, and in fact go up an entire new stage, that 6 months is a critical window of opportunity where if we can provide an answer through a non-invasive or minimally invasive liquid biopsy assay, that's much better for the patient, and it's much better for the clinicians who have to care for those patients. And so, to summarize, where do we see this fitting in? We see that we want to use our assay to help clinicians rapidly address the question as to whether or not a detected nodule, again, whether that's incidentally detected or through a lung cancer screening effort by a low-dose computer tomography, whether or not that nodule is malignant or benign. And so we've been working on this now for 2 years. We've looked at a very large cohort of patient samples, over 1,000 lung cancer and non-cancer lung disorder samples, and we are sort of in the final stages of transferring that assay to our CLIA laboratory, where we hope to offer the assay to clinicians next year.
Thank you, Eddie. And then Greg, in which ways can ctDNA and microbiome liquid biopsy tests be combined to enhance the detection of tumors?
It's a great question as well. And I think, as Eddie had said previously in this conversation, the microbiome is really completely a human genome independent source of diagnostic information. So the opportunity here just makes a lot of sense to synergize both the human component, which people have been using and exploring for the last 10 years or so for cancer diagnostics, and pairing that with microbial information. And because they're orthogonal in a sense, we think that the benefit from both is going to provide this great diagnostic synergy. So the performance should be better than either of them individually by combining them. And so yeah, we're actively working on opportunities in order to take the human information that's readily available in blood to improve what we already consider to be a great diagnostic just using the microbial information. And as we continue to do this, we will build what I consider to be multi-species, multi-omic tests, where the multi-species is the microbial and the human component. And I think this is truly the way to move forward to create the best possible diagnostic. Because at the end of the day, what really matters is trying to help patients like my grandmother not have to be diagnosed with stage 4 disease. Whatever information that we can put together in order to make an earlier detection is what's ultimately going to be beneficial to both the patient their families and the physician that's in charge of taking care of them. So yeah, it's a huge opportunity. We're actively working on it, and yeah, we're excited about what's to come.
Thank you. And you've touched on this slightly. So what are the sort of next steps for your research, and how would you like to see it applied in cancer diagnostics?
Yeah, we have talked about this a little bit. I think there's a major difference in the scientific literature between something that's just been published, even in high impact journals, and something that's actually been validated clinically speaking. And this involves prospective clinical trials, right, of diagnostics with hundreds to sometimes thousands of individuals. And so what we've done in the past and what we've published has provided a lot of support that we're on the right path. And we've been working on this more than 1,000 patient lung cohort with also validation in a completely blinded set of individuals with very, very early stage cancer versus those that have lung disease. And we've been excited by that because it's shown that our diagnostic is currently doing better than the state of the art, but much better actually. I think when the rubber's going to meet the road is really applying this in prospective trials and showing not just how well the diagnostic is performing, but how much are we actually going to help patients. And all of our models are suggesting that it's going to be a huge improvement, but it's going to take time and a lot of effort. And that's the exciting part of what we get to do, is really trying to translate this to the clinic. And so yeah, that's what I'm really looking forward to.
And then Sandrine, could you talk us through Micronoma's current research pipeline focusing on early cancer detection in microbiome-driven liquid biopsies?
When we do research and development. So on the development side, just to echo what Greg was saying, there will be a lot more at-scale prospective study validation for the early cancer detection of lung cancer. And the first part is going to be supporting what Eddie was mentioning, the utility of this tool to the clinician, to the payers in terms of reducing costs and delay in treatment for lung cancer when in the context of newly discovered, incidentally discovered nodules. So that's going to be the first development. The second logical development is that in US, for example, we know there is 15 million people who are at risk of lung cancer. They are the 50 years old and above and heavy smokers. Right now, they are channeled through the screening standard of care, which starts with a low-dose CT scan. There is not enough of this low-dose CT scan done. It's around 4 to 5% of the people who should get their low-dose CT scan get it, which trigger into lung cancer being detected so late. So the goal for us is going to, at some point, once we are done explaining the utility to use a technology, cancer versus benign nodule, will be the next logical step, which is, okay, well, let's do cancer versus an otherwise healthy person in which we don't even know there may be a nodule yet. And so to channel more readily people to do through that screening and saying, hey, careful here, there is a red flag. You're in the at-risk population. Now we have a blood test to tell you that you should really, really go through the screening process. So that's around the lung aspect of development. Then going back to the R of R&D, the research aspect, we're going to continue developing and researching this methodology in other cancer than lung cancer. Greg was mentioning, losing his grandmother from pancreatic cancer. I happen to lose my father-in-law to pancreatic cancer as well. So obviously, this is one of these cancers that unfortunately is detected too late and is too deadly. So cancers such as pancreatic cancer, ovarian cancer, liver cancer are definitely part of our pipeline, because as Greg and Eddie was mentioning, this is not unique to lung cancer, this microbiome oncology relationship. It links to multiple cancers. So we are going to go to this pipeline to enable everyone to get screened or detected, diagnosed sooner, so that there is a better chance for an efficient treatment.
And what are the other applications of microbiome-driven cancer diagnostics?
Yeah, so that's the important thing. So we decided to focus first on early cancer detection. But, unfortunately, we won't be able to detect everyone early just because they may not be on time for that cancer test. So developing this technology for minimum residual disease detection or relapse detection is going to be on our pipeline as well. One topic we didn't touch too much on, and I'm going to do a bad job summarizing some of that work, but more or less finding the tissue of origin of the metastasis is super important, because you don't treat a primary cancer the same way that a metastatic cancer. And it seems that metastases, cancer cells, when they are metastases, may carry their load of microbiome with them, enabling potentially us to detect back and track back what was the tissue of origin to help clinicians make the ultimate decision around treatment. So these are the application of that within the past of treatment and diagnosis. Obviously, someone is going to find a mine of new pathway to help. It's going to be challenges, as Greg was saying. We're not looking at one or two bad mutations. We are looking at multiple bad agent or good agent. That's another webinar, I'm sure. But that will be a mine of potential new therapeutic pathways. But going back to existing therapeutic pathways, there may be a drug that has been failing in clinical trial that could be quote unquote rescued by doing a rich stratification of the patient who are in the clinical trial on high responder versus low responder based on their microbiome profile in their blood. And the good news is that we are looking at plasma, and a lot of this trial have plasma banked. And so we just need to go back and remind all of these samples to figure out if we can find a classification tool, a stratification tool, to rescue some of these drugs and put them in the hand of the clinicians for the patient that would be respond to that. And so that would lead ultimately to new companion diagnostic application. So early cancer detection is really just the beginning of what can be done with these platforms.
Thank you, Sandrine. And then I have a question for everyone. So to wrap up our coffee chat, could you briefly each tell us how you'd like to see the field progress over the next 5 years? Greg, do you want to kick us off with this one?
Sure, I'd be happy to do so. I think what we've seen over the last 3 years is an emerging picture in which you have a dynamic community of bacteria, fungi, and viruses all being present in the tumor at the same time as cancer cells and immune cells. And a great question that this all begs is, what is happening to the cancer's evolution? And I think on the human side, people have worked very hard over the last 5 to 10 years in order to develop clonal evolution models about how cancers evolve over time and eventually lead to treatment resistance and or metastasis. And unfortunately, none of these models, to my knowledge, have actually included microbial information. And this isn't simply, just let's include it for the sake of including it. Like, we're talking about, it is already known that bacteria affect every single aspect of a cancer cell state. From the genome, it can create mutations. From the transcriptome, it can rewire programming. From the epigenome, it can cause epigenetic modifications. To the metabolome, where we know that bacteria can exist intracellularly and actually modify the metabolites. For example, there are about 300 species of bacteria that can degrade gemcitabine into a non-functional byproduct. And many of these bacteria have been found intracellularly in the case of pancreatic cancer. So you're effectively talking about a new cancer clone being developed by the infection of this cancer cell by one or more microorganisms. And this is also probably true if these microbes are spatially adjacent to this cancer cell, but even not necessarily inside the cancer cell. And I think on top of this, if you consider how microbes are spatially distributed within a tumor, which the data is preliminary, but it already suggests that it's not homogeneous. So microbes prefer certain regions of the tumors than others. Well, it's all the ingredients that you need for effectively modifying clonal evolution. And so I think one thing that's really exciting to me is, how are we going to now consider clonal evolution as a multi-species process? And I had the opportunity to write a review paper essentially proposing some of these hypotheses that we've had. And I think it's exciting to me to see how the field will validate or potentially reject that theory, but I think there's a lot of support going forward. And the last thing that I'll mention, of course, kind of bringing this home, and I've mentioned this a few times throughout this chat, is making papers into real world translational diagnostics that reach the hands of patients, that reach the hands of clinicians. This is a really, really difficult effort, that is unfortunately not common enough in academia. And I'm really excited to be able to translate these academic findings that I made during my PhD into these real world solutions that can benefit patients. And so that's probably one of the most exciting aspects for me.
Thank you, Greg. Sandrine, could you let us know how you'd like to see the field progress over, say, the next 5 years?
The research is there, it's burgeoning, there's so much more to develop, but what I really hope is that it's going to go in the hand of the clinician and the patient sooner rather than later. And from a non-scientific point of view, the challenge is nearly as great as a scientific point of view, because it's going to be all about adoption. So number one, we need the clinician and their patient to understand that we're not doing something weird by, asking microbes to tell us about the cancer status. Microbes were there before the humans were on the planet. And so we're just mining something that should have been obvious in a way to all scientists following the timeline of evolution on the planet. But we still need some conviction. I mean, we are detecting cancer with microbes, except Helicobacter pylori or HPV. That was not really well known. And now we are not just saying it's one microbe or two microbes. It's 50, 100 microbes that we are mining to get that. So the education and the comfort of these things, saying, no, it's real. It's not snake oil. That's going to be one big focus. But we have the support of all of the science coming out. The other thing is that we can develop the best diagnostic technology. If no one can afford it, no one is going to use it. And all of our hard work on the scientific side will go nowhere. So a big focus will be to help everyone understand, including the insurance payers, that there is humongous cost saving in using this kind of solution, because early detection means early treatment, meaning better chance of survival. And all of that is, number one, costing less to the healthcare system, and helping everyone to continue living productive life instead of succumbing to the disease. So to me, these are going to be the main focus on the 5 years, next coming 5 years, is just making sure everyone understands that we are a mine of information, and then making sure that the clinician and the patient can afford to use that solutions.
Thank you, Sandrine. And Eddie, could you let us know how you'd like to see the field progress?
So there's really three things. The first, echoing sort of Sandrine's comments, is, and I think will be something that Micronome will play a large role in, is really driving the clinical adoption and utilization of a microbiome-based liquid biopsy assay for early cancer detection. I think that's a real game changer for many of the reasons that we've outlined today, and I think that's going to be extremely exciting for us to play a role in, but then also to see how the field overall responds. That's going to be an extremely interesting thing to witness over the next 5 years. And then secondly, just reflecting on the things that we've done in the development of our lung cancer assay, I'm very excited at the prospect of, in recognition of the diversity of microbial signal that we find in plasma, in circulation, really starting to see the development of more multi-omic diagnostic technologies in which the microbiome plays a very important sort of foundational role, where you can imagine taking a layer of microbiome features on top of which you might add proteomics, metabolomics, clinical data to really make very, very high density or high feature set diagnostics that can leverage the power of machine learning to arrive at a really sort of a next generation of diagnostics, very unlike what we see in the marketplace today. And then lastly, which sort of stems from my interest or my training in immunology during graduate school and my postdoc, I was doing work in immunology during the early stages of the discovery of the toll-like receptors. And, it was, at the time, it was appreciated, or it was shown that certain motifs within bacterial DNA are very, very potent immune stimulators. And what's very interesting, and that I'm very eager in sort of seeing how this research unfolds, is that we see abundant microbial content even in healthy individuals in plasma. So the question is, how is it that, given this sort of this historical immunological data, that bacterial DNA is a potent immune activator, how is it, what mechanisms are at play that allow microbial DNA to be in circulation in the absence of immune stimulation? So I think that is going to be a really fascinating piece of biology to uncover, and it's really exciting to know that those discoveries will be stemming from this work in cancer diagnostics using microbiome-based platform.
I think looking towards the future is a good place for us to conclude this coffee chat. So I'd like to thank you for your conversation today and for highlighting how the microbiome can unlock a plethora of options for pushing forward cancer diagnostics. So thank you to each of you for joining us.